Are you recording?
We will start right now to record. And also we're going live on the MARC Facebook page.
Mm-hmm.
Okay. Would you please start, Dr. Samira, with the introduction?
Okay. So, hi, everyone. It's our pleasure to welcome you all in our monthly webinar. So just to give a brief introduction about MARC, Munir Armanius Research Center. It's a newly established research center that aims to translate scientific discoveries into effective medical practices. MARC is performing its operation in 4 unique areas. A first clinical operation, including clinical trials that evaluate medications and real-world data, research projects we will be doing and using latest omic technologies, aiming to improving patient care and supporting medication production, and will be having a genomic and biorepository facility in the bioequivalence and phase 1 trial center. And recently we had a call for proposals that was announced in September 2021 for project based on innovation and excellence. This grant is awarded to high priority basic biomedical research project with the potential for high scientific impact and high user impact, addressing new concept and technology to improve health. And in this call for proposal, we focus on 3 areas, mainly immunology in cancer and cancer research, non-alcoholic steatohepatitis and multi-drug resistance bacteria. And we received a lot of application, and all the application were reviewed by external committee. And now we are in the process of releasing the results. In today's scientific webinar, Dr. Katalin Kariko is a world expert biochemist and a pioneer researcher who devoted her whole career to uncovering the potential for messenger RNA therapeutics, takes us into a journey of messenger RNA discovery and its wide potential for applications. Dr. Kariko, hero of the year 2021, contributed significantly to the breakthrough development of the COVID-19 messenger RNA vaccine, fighting the pandemic, and now the messenger RNA therapeutic is paving the road to many healthcare solutions. Dr. Kariko is a senior vice president at BioNTech and adjunct professor of neurosurgery at University of Pennsylvania. She joined the Perman School of Medicine at the University of Pennsylvania in 1989 and began collaborating with the American immunologist Dr. Drew Weissman in 1997. Her research has focused on RNA-mediated mechanisms with the ultimate goal of developing in vitro transcribed messenger RNA for protein therapy. She investigated RNA-mediated immunization and co-discovered with Dr. Drew Weissman that nucleoside modifications suppress the immunogenicity of RNA, which has widened the therapeutic potential for messenger RNA in treating different diseases. This led to the development of 2 most effective vaccines for COVID-19, the BioNTech Pfizer and Moderna vaccine, and she holds a wide promise for future treatment for many other diseases. She received more than 50 awards in the last 2 years, including the Breakthrough Prize in Life Sciences and the Princess of Astras Award and the Violet Prize for Excellence in Biotechnology. She continued to work in a new therapeutic application of messenger RNA therapy. Her specialization in messenger RNA-based gene therapy, RNA-induced immunoreaction, molecular basis of ischemic tolerance and treatment of brain ischemia. Together with Weissman, she holds a US patent for the application of non-immunogenic nucleoside-modified RNA. This technology has been licensed by BioNTech and Moderna to develop their protein replacement technology, but was also used for their COVID-19 vaccine. Thank you very much, Doctor, for accepting to be here with us today. And we know how busy you are, but thank you. We appreciate your time.
Thank you. Thank you very much, Samira, for this nice introduction. Let me share the slide here. Okay. Is it good? Mm-hmm.
Yes, doctor.
Yes.
But you can see on the side notes also?
Yes, we can see the side notes. I think we can press the full screen button.
Yeah. I tried that. Okay.
Okay, that's perfect now.
Yes. So thank you very much for inviting me. It's really congratulations to this center, and I wish good research to be done there, and great phase 1 trials that you are planning, and a lot of immunology and so on. And so that's really is true that wherever is knowledge is accumulating, that invention is coming. And you know, from the old Egypt, it is true that, you know, there were the libraries, there were the information and how you proceeded. And today we are here talk about messenger RNA. And I will talk about, you know, what was the past, how we get where we are today, and also that what seems that the future holds for the mRNA. And of course, all is started, you know, when the mRNA was discovered in 1961, and it took like 60 years to have the first mRNA-based product ready. And so during this 60 years, so many things happened, and I most of the time I was around, not 61, and I couldn't even read in Hungarian, not that English, to read those papers, but this progress was usually an incremental progress when the mRNA was developed product. And so, as I mentioned here, I put several references here for all of the audience that who later want to check it out. This is a very good recent review, and actually it includes many things which I am talking about today, because the mRNA development for therapy was depending on constantly improving the mRNA. That's the left side, this red arrow, and the blue is the delivery, the lipid nanoparticles. And so it is continuously one or the other was advanced, and then it helped always that we could get better and better results. So I am recommending you to read this paper. It's a very high quality recent publication. So the RNA was discovery was described to Nature paper, and then already scientists were looking for, well, how could it be that, you know, translation is in the ribosome, and all of the genetic information by 53, we already knew that in the DNA, in the nuclei, and they couldn't find in the cytoplasm where the protein synthesis occurred, they didn't find RNA, and eventually they found that this intermediate, and of course, most of the papers said unstable because it was so difficult to isolate, especially in bacteria, where it, as an RNA is made, translated, and then almost immediately degrades. But of course, you know, the scientists, at the beginning, they try to analyze how this RNA looks, what is this all mRNA is all about. And in 1975, they discovered an important element on the 5' end. And in that year, several Nature papers reported, you know, that this interesting so-called cap structure is there. And already in Furuichi, Shotkin, when they showed already that when they remove this element, translation is not occurring. So it is very important for the rest of the story that we have a very good cap on the messenger RNA. It was by the end of the year, Bernard Moss and NIH, they also demonstrated that there are these enzymes which can cap the mRNA. And it was also important because many times we did use these viral enzymes to cap our RNA for our project. And why scientists, you know, try to analyze the RNA, which is viral or naturally occurring in the mammalian cells, other scientists isolated RNA. And they went and isolated the RNA from reticulocyte, which is enriched in globin RNA. They couldn't synthesize because we didn't know how to do. The molecular biology in the 60s, 70s was not there to help us. And this RNA, because this is the rodent reticulocyte so overwhelmingly enriched for globin mRNA, so they could separate and isolate. And in the 60s, they translated in a reticulocyte lysate. Later on, they injected in frog oocyte. And there, here in 1978, two Nature papers reported that they put in liposome and then they fused into primary cells. So actually, this was the first mRNA therapy experiment. And they, after this process, they demonstrated that the murine lymphocytes was making the globin, the protein. So when this we read, actually in 78, we read this paper, we get excited, and I started experimenting with RNA and liposome. But here, actually, we just started with liposome and we delivered DNA, not RNA yet, because we did not isolate it and we didn't know how to synthesize. And these colleagues already started, Eva Kondorosi, Ernő Duda, in this biological research center in Hungary. And they already put into liposome nuclei and viruses, and they delivered to the cells. And it was, this fusion was so magic. And so as a student, this was my first experiment as an undergraduate student. And so I can recommend, if students are listening, that doesn't matter what you start with, you just do an experiment and make your hands wet, and then start to think and learn about, because this first experiment was very decisive for me, and many times I thought about that. I actually eventually went to work with RNA in the same center, and then as a PhD thesis I worked with small RNA. But meanwhile, while we were doing a different experiment in Hungary and later in the United States, 1984, an important advancement was in our field of the mRNA therapy field, because Douglas Melton, Paul Krieg at Harvard University, they described how to make RNA in a tube. So scientists even before them tried bacterial RNA polymerase, but it was, it was not very reliable. And here turned out that it is high fidelity RNA could be generated. They made it human beta interferon coding RNA. The RNA has a 5' UTR structure, 3' UTR poly-A tail, and as I mentioned, the cap structure. And this actually, they meet with the capping enzyme with Bernard Moss, who just described 75, that viral enzyme, they added it. And this mRNA, they injected to the frog oocyte. Then those oocyte, they cultured in medium, and they demonstrated that the protein which was made is functional, and it inhibited viral replication. So this 1984 was also a very important advancement, because now that we could demonstrate that on design, we can make an RNA and code for a protein which is translate functional in a cell. The subsequent years, between the 90s and first part, scientists get excited about using this in vitro transcribed RNA, and they inject it to animals. They did studies where for protein replacement, like vasopressin mRNA was injected to brain of the rats which suffered the disease, and they could see improvement. 93, 94, two papers describing how mRNA can be used as a vaccine and against influenza. And the first paper by Martin and Meulian, they actually used liposome. They put the mRNA coding for the viral protein. And after that, you don't see many of the infectious disease related vaccination. Mostly the field moved to make a vaccine for cancer. And these were injecting the RNA to the naked RNA to the mice, or Eli Gilboa at Duke University. He did ex vivo experiments. He used the isolated immune cells, human dendritic cells, and delivered the mRNA. And actually, he did establish the first RNA, messenger RNA company called MARC in 1997. And he advanced the messenger RNA all the way to human clinical trials. And this company became later Argos, but they treated the patient sample ex vivo, so not injected the RNA to the patient. And so what was the problem? That we could see only one paper per year and not much follow-up. And that is the problem, is this very small amount of protein was made from the RNA, very small amount and very short time. So why? Because when they inject the RNA, it very quickly degrades. And the translation is not very effective, because different things which should be present in the RNA, we couldn't do it. The RNA also was inflammatory, and so many shortages, So most of the people were not interested, especially if I think about those laboratories who work with DNA. And if you work with the molecular laboratory, use plasmid. When you isolate, you start with like a cryogen kit. You add the RNAs, and then you vortex, and then you already contaminate half of the laboratory with the RNAs. And so it is very difficult to work with RNA in the laboratory where people are working with RNAs. So that was the major problem. So for me, I started to work in the 89 with the messenger RNA. And for me, as I mentioned this, how the formulation and the RNA advancement of those structure and delivery method helped advance our field. So for me, the lipofectin, which is a positively charged molecule, and helped to work on culture cells, because we just put together the RNA and the lipofectin, which is RNA is negatively charged, lipofectin positively charged, and then we deliver to the mRNA to mammalian cells. Working in cardiology, we were interested in urokinase receptor, and to our surprise, this highly post-translational modified protein was made. So the cell knew what to do. We just had to deliver the mRNA. And later, moving to neurosurgery, we tried to do mRNA, which coded for different antioxidant enzyme and so on. We did in vivo experiments and many other things. And what happened is that I did not see any problem with the RNA. My major goal was to deliver an RNA coding for a therapeutic protein, and the protein which is normally present in the body, but not enough wherever I wanted to deliver. And then so I just helped the nature, because after all, we did not invent messenger RNA. Nature invented, we just tried to use and help with it. But what happened is that when I met Drew Weissman, my colleague, as you just mentioned, and he was interested in messenger RNA. He was interested in to develop vaccine, and he said that he wanted also tested out messenger RNA as a vaccine, but he couldn't find anybody who would have that. And we just met accidentally at the Xerox machine, and then I offered my help. And so what happened is that we found that the RNA I made, it was so inflammatory, and which is not good, because for me it was not good, because I wanted to use to treat a patient who has stroke. And although Drew was happy, because he recognized that all of the important factors were upregulated in the dendritic cells, which required for the good vaccine. But this inflammatory response, the high level of TNF-alpha, which is induced by this RNA I made, this is the purple bar here, was unfavorable. And we were thinking that why why it is that the RNA is inflammatory. Could it be because we are adding the RNA from outside to the immune cells? And based on it, maybe all of the RNA, which is even inside the cell, could activate and induce inflammatory molecules. So we isolated nuclear, cytoplasmic, mitochondrial, and so on, different RNA out of the mammalian cells. And what we found is that they were inflammatory, but not as much as the in vitro transcribed. And what we found is that the tRNA did not induce any TNF-alpha, this very inflammatory cytokines. And knowing that the tRNA has so many nucleoside modifications shown here, like 25% of this modification, 25% of the nucleoside is modified. So one thought was that maybe that's the reason, maybe the modification, but of course it is shorter than the other RNA, so it could be that is the reason. So you have to know that naturally in your body, the RNA is made from the 4 basic nucleotides, always, whether it is tRNA or messenger RNA, but post-transcriptionally or co-transcriptionally, where it is still making, there are different enzymes introducing these changes. But when we figured out that we should make an mRNA containing these different kind of modification, we did not know that what are those enzymes. It was not that commercially was not available, but of course it was not even known what are those enzymes. So we were thinking that how we could prove that this is really the case. So we had to purchase already nucleoside triphosphate, which already modified, and these are listed here. And we try to see that whether with a polymerase, phage RNA polymerase, we can incorporate. And we use in this experiment, as well as for the vaccine and the many others, is the T7 phage RNA polymerase, and which has a very short promoter. And then we try to see that whether it incorporates, not all of them incorporated, but some we could see that it incorporated, and we could generate an RNA. So the next question was that whether we reduce the immunogenicity of such RNA. And in purple or in blue, actually here, you can see that the RNA, which I synthesized without any modification. And what we found is some of them which contain modified nucleoside, and those were not inflammatory. And when we took a closer look and see that what are those who don't induce TNF-alpha, actually they all had modified uridine. So it seems that the nature selected uridine to recognize as an external RNA, as an invading RNA, and reacted it and induced inflammation. But to proceed for our final goals, Drew Weissman to develop a vaccine coding for HIV-specific proteins, and me to develop RNA for therapy, it was important to check out whether RNA is translatable, and that's what we found, that the RNA, when it was M5U or pseudouridine modification, it translated very well. Actually, for pseudouridine, it translated 10 times better than the natural RNA, the uridine-containing RNA, and we spent like 2 years trying to figure out why. I might mention this modification all naturally present in our RNA. So pseudouridine, actually the 5th most abundant nucleoside in our body. So these are natural molecules. At this point, we are in 2005, we didn't know that messenger RNA has pseudouridine. It was 2014 when demonstrated that mammalian and other messenger RNA also has pseudouridine present in them. So when we did testing, we realized that our RNA is not clean enough. because during the transcription, we get unwanted products. Those were double-stranded RNA, which inhibited translation. So as I mentioned, in our goal on this field was constantly increasing the translatability of the RNA, better delivery, cleaner RNA to produce. And we used the HPLC purification, and here using a J2 double-stranded RNA-specific monoclonal antibody, we could demonstrate that the condition we found, and again, it was like working on for 2 years, finally identify a condition how we could purify the RNA. You know that there are, after HPLC purification, no double-stranded RNA was present, and we could see that it greatly increased or actually the purification greatly reduced the inflammation, which could cause by these RNA. Here you can see different interferon and interleukin molecules. And although the unpurified RNA, when contains modification, it was less immunogenic than the uridine containing RNA, but after purification, even we eliminated those remaining inflammatory molecules, and the translation increased tremendously. Here on the right, you can see those with the blue, those are purified, and especially in primary cells, we could get great improvement, several log better translation when we tested out these purified RNA. After that, was important for us to see that now that we can make purified, modified RNA and whether it is translating in vivo. And for that one, we selected erythropoietin, which is a hormone that stimulates red blood cell differentiation. And as a potential treatment for anemia would be if the mRNA translates well and erythropoietin is present for several days. And we selected mice and injected with the murine EPO mRNA. And very small amount. If you work with RNA, you know that 0.1 microgram is a very small amount. And when we injected pseudouridine containing RNA, and 4 days, we could measure erythropoietin circulating in the blood. Actually, some experiment when we measured even 15 minutes following injection of the RNA, we already could measure the EPO erythropoietin in the blood. But when we had the conventional RNA here in blue, then the RNA did not translate it as well, disappeared, and we could not see more. And here is the protein, which usually has a 6-hour half-life. The RNA was functional. The RNA, I mean the protein generated from this injected RNA was functional. Here in the red, you can see that hematocrit increased in the animals, so that more red blood cells was produced. And when we weekly injected the RNA, that we could maintain high hematocrit. And of course, the most important, I'm saying last, is that the RNA was not inducing interferon. It was not immunogenic. While we were doing all of these things, and we are now in up to 2012, and continuously improving the RNA purification, different elements we changed, we modified it. Here in Germany, my colleagues, Ugur Zainin and his team at BioNTech, they did a lot of improvement as well. They did improve the cap analog, they used the thio cap analog, they improved the untranslated region of the molecule, incorporated different sequences, which helped to use them as a cancer vaccine. And their study advanced so well that by 2012, they started human trial injecting the RNA. So it is not ex vivo, the RNA was injected. And here by 2017 here showing the data when individualized cancer vaccine was produced and showed a very effective in the patient now. And they also here made a progress at BioNTech, where formulated the RNA with lipoplex, how we call it, and found the ratio when positive and negatively charged ratio was changed, same composition, but injecting the animals, going to in the spleen or going to the lung. And so this advancement was made by us at the University of Pennsylvania and by biotech scientists. And really what happened, that what I showed you in the 1990s, we had just a little bump, little protein was made short time. Now that we had significant improvement, and now that we dispurified and coding segments optimized and so on, we could have a significant amount of protein made. So I came here and joined BioNTech in 2013, although at that time we didn't have this fancy building. We were still on the campus, and not even we didn't even have a website. And what we have done here, further improvement of the RNA. It is just an endless process, and also many processes which we could do at the bench. We had to come up with a solution when it is scalable, so industry could use it and available, because we precipitated the RNA and then you just cannot use it in large scale. So one of them was codon optimization. I might mention about codon optimization that some others are using to reduce uridine content by selecting G series sequences and doing other optimization to get more protein. But I myself is very concerned about codon optimization, because immunogenicity of the mRNA in that time will be dependent on what will be the amino acid composition, because there are amino acids which are very frequently present in the proteins. They require uridine 1 at least, or in case of phenylalanine at least 2. And so every time the RNA immunogenicity depends on the composition of the protein, what kind of amino acid are there. In addition, if you are using RNA for vaccination, you have to be aware that many times under stress, out of frame translated products are also important immunogen antigens, and you can lose it if you optimize, and or you can create some new product by codon optimization, which will be irrelevant, and you are just generating unwanted response. We did a further improvement when we recognized that how important is CAP1 presence in the RNA. So usually using the non-reversible CAP analog incorporating during transcription, we get quite high translation early on, but it dropped. But when we use the CAP1, in that case, again, we use erythropoietin mRNA, we can continuously translate it. And the reason is, as is described in this publication, that when RNA is delivered to the cells, the cell membrane disturbance result that the inhibitor molecule, so-called interferon-induced tetraticopeptide, is induced, and this is a blocker. It can block, but only it can block the CAP0 RNA. And so if we have this methyl group present in the first transcribed RNA, then this cannot bind to it. It blocks the inhibitor binding, and only the translational factor, eukaryotic initiation factor 4E, can bind. And then we get very good translation, as shown here. We did a further purification. I mentioned the HPLC, which we introduced in University of Pennsylvania. And here with my colleagues, we did introduce a very simple method using cellulose to remove double-stranded RNA from the product. There is the RNA polymerase, this phage RNA polymerase, has an effect of unwanted RNA-dependent RNA polymerase activity, and during transcription, we get these double-stranded RNA product. And when we develop this method and we can purify the RNA, it increased the translation of EpoRNA tremendously. We know why we have now RNA which is purified and optimized in many different structural elements, the cap, the polyether. We started to use it first in animal studies. Here we describe how we generated a bispecific antibody, and which helped to eliminate large tumors. So the bispecific antibodies can see in T cells and can see the cancer cell, and that can bring it close proximity. These bispecific antibodies are in clinical trial, not this one, but others. But the major problem is that the half-life is very short. So the RNA, we could inject just weekly to the animals, and it produced enough protein that it lasted much longer, whereas the protein, bispecific antibody protein, had to be injected more frequently. And right now in patient also, that every second day they have to visit the hospital to fill up their peristaltic pump, which inject the bispecific antibody into their bodies. Last year, we published another application. Again, this is nucleoside modified RNA. When we delivered this mRNA coding for autoantigens and demonstrated in mice that two different models of multiple sclerosis, that we can induce tolerization against the autoantigen. And so here, the difference is that the tolerization and the vaccination, which we did with other vaccines, is that here the RNA was non-modified and the lipid did not induce any response. And this has not advanced to human trial yet. We need more time for that. And of course, now here we are, when development of vaccine for SARS-CoV-2 occurred, and this you are very familiar with how it happened in 2020 January, when the information of sequence information became available, and 150 or 200 different places scientists started to make a vaccine. And I might mention that if this pandemic would happen 20 years before, scientists who want to develop vaccine would need a material physically in their hands, some viral material to start with working. But now information was sufficient because gene synthesis was already established and a lot of progress in molecular biology occurred. So as you know, by end of the 2020, we had vaccine was approved, and last year, 2021, our vaccine get the final approval. A lot of people at that time thought that, oh, this was the first time actually this nucleoside modified RNA was used, not just this vaccine, but also in the Moderna vaccine. But those who are familiar with the field knew that actually other clinical trials were already ongoing, where a nucleoside modified mRNA was used for therapy. This it was described first in 2013 that VEGF-A mRNA, which codes for a protein that helps to generate new blood vessels, when they inject it to animal model in mice suffering in heart failure, they could improve the performance of the heart. And this was entered to human trial, and actually two different trials. One in which wound healing was accelerated by VEGF-A, and this started 2019, and 2018, which was patients were injected in the heart. And these are patients who are undergoing elective coronary bypass surgeries, and VEGF-A mRNA, naked mRNA, was injected in 30 different places into their heart. And if you watch out for news in these days related to the RNA therapy, in this year, January, was announced that those patients who were treated in this way with injected VEGF-A mRNA, then their heart performed so much better. And on this video, you can see the report with them. So that was already a successful use of nucleoside modified RNA. Last year also we heard about that Intellia was using the Cas9 mRNA. It was a nucleoside modified RNA to treat an amyloidosis. It's a transthyretin amyloidosis, and this is a very serious disease, as you can see. What they did is that they delivered in a lipid nanoparticle the mRNA coding for a Cas9 enzyme, which was guided by this guide RNA to the problematic part of the genome in the liver. And when they delivered to the patient, the RNA was translated, and the Cas9 protein, which is associated with the guide RNA, entered to the nuclei on the patient liver, and then interrupted production of a very toxic misfolded protein. Again, we just heard recently, a couple of weeks ago, that the patient who received this kind of treatment, they even today, after 1 year following the therapy and only one injection, so that this is permanently changed their disease, treated the disease, and they were still not making the toxic protein. So we also did a study, as actually one of the first one that I also participated to initiate. And this was when we delivered messenger RNA coding for 4 different cytokines. And the experiment is that when a cell surface tumor is injected with this kind of mRNA, the tumor cells are translating, and the generated protein actually invite immune cells to this lesion, to the tumor. And then they are activated there. So the cold tumor became hot tumor, and then the circulating immune cells will eliminate remotely located tumors. And on the right, you can see that the control RNA, the lung is a melanoma, and the control mRNA treated animals had a melanoma in their lung, but not in those who were treated with this cytokine mRNA mixture. This project moved to clinical trial, and together with Sanofi, and now patients are treated with such mRNA mix. I can keep on going and ongoing and telling you about how many preclinical studies are at present, and actually daily we heard about, even today I was reading that a new clinical trial is started with using nucleoside modified RNA, and in these days mostly infectious disease, which was for HIV. Moderna initiated 2 different HIV vaccination trial, Zika virus, CMV, RSV. Those RSV is already very advanced in phase 3 trial, flu, and of course more COVID related. And for cancer also is used both nucleoside modified, and there is also unmodified RNA, and those are whether intratumoral injection or injection of mRNA coding for the antigens. And the preclinical studies, in a lot of preclinical studies are ongoing in animal studies, and from treating edema, bone healing, and many, many other things. So just to leave time for questions, I might just stop here now, and I hope with all of the references, if you are interested, you can extend your knowledge on this field. And by that, thank you for your attention.
Thank you very much, Professor Kariko, for an overview, excellent overview of messenger RNA advancement since very early. So it's now clear that this new science is not very new. It has been for a while. Just maybe the COVID has put more attention and light into it. So now we can open the questions. So can you see the questions or in the Q&A section?
Yeah. Yeah. Please, why mRNA need a special storage at minus 80 degrees? Why siRNA just being stored at refrigerator? So first of all, I would say the siRNA personally is not an RNA. You know, in siRNA, 2' doesn't have an O, it is 2' fluoro, all of the pyrimidine. The purine has 2' O methyl. The RNA is short and double-stranded. Even you can inject to the blood, it won't degrade that easily. The messenger RNA, you don't have to keep it minus 80, by the way, and it is not about RNA because the vaccine is lipid formulated, you know, so that it's not the same. The case of siRNA, which is now it linked with GalNAc, is not in a lipid, no? But what happened is that we had data for minus 80 Celsius stored lipid nanoparticle formulated mRNA that even 6 years later, those were of course erythropoietin and other different reporter related product, but we knew that it was for years is stable. And so that why we started with minus 80, because we knew that. And so you have to think also that when it is minus 80, you don't have to go through your freezer every 6 months or every 3 months and throw away everything because, you know, you are keeping in higher temperature. You know that this also, you can keep this RNA for a couple of days, even on room temperature, and you can inject. Again, you know, that you have to prove and you have to provide data. And that at the beginning was you needed time to generate with the COVID-19 specific sequences. So it is, the RNA is inherently labile. You don't even need an RNase, it can degrade. You know that you never heard about that, oh, the dinosaurs, they just dig out and they found RNA. It is always DNA because that extra hydroxyl makes it so labile. So you can keep it lower, higher temperature, just shorter period of time. And remember, the vaccine is not just the RNA, it has the lipid nanoparticle. Is there, now here is Fatma Khalil, is mRNA targeted to viral encephalitis or HIV? I mentioned that Moderna initiated 2 HIV studies, one just recently against, together with NIH, so, and different construct coded by the mRNA. And viral encephalitis, I am not aware of that. Only neuronal related is the herpes. And those are not initiated yet, but it was prepared. But you heard that both Moderna and BioNTech Pfizer reported that they will, in the second part of this year, they will do shingles, which is a herpes virus. And there are side effects of mRNA targeted anti-tumor. What are? Probably what are? Oops, what are? Which one? Oh, okay, something disappeared. Okay. Okay.
What are the challenges you faced during your transition from academia to industry, and what's your advice for young scientists considering the same steps?
Okay, so I worked up until I'm 58 years old. I worked at academia always, and I found uplifting to come to the industry. Of course, what was beautiful is that what was the goal is to make something which is helpful, helping somebody. You didn't have to make another paper, and nobody cares just because you need your CV to look good, but you needed to make a product which helps somebody. And that was so uplifting. And the other is that in academia, I found very competitive, you know, you try to work with somebody, but then they are competing that who will be the first out or the last out or something like that. And here it didn't matter. We have to have a product. Everybody worked together. So I found this very uplifting. And of course, you know, I mean, I am from Hungary, and I experience what is there, what is in the United States, what is here in Germany. So, so if you have an idea that you are the best person who work on it, you have to, you know, get some money from somewhere, spin out a company, and you go after it. That's how I can see and suggest to other people that who say that, oh, we should do that and that. Oh, okay, if you think that way, then, you know, try to pursue that. And so that's, for me, my experience is that it is not for the money. People just so, I don't know, they think that if you are in an industry, I mean, the industry won't have the vaccine, won't have that and other things, you know, we were sitting at home. So you need somebody to produce those things and come up with ideas. And for me, actually going to the United States was the biggest impact on me, was that at the University of Pennsylvania is an Ivy League school. And so good presentation, I could listen, you know, great scientists came for all over the world. But today you turn on the YouTube, you know, anywhere in Cairo, you can listen to everybody, you can learn. You don't have to go there, you know, and so that's very important to keep up your knowledge up to date and different sites where you can follow the latest presentation. And I encourage, because that's what I was doing and still I am doing. And now I'm 67 years old and I still try to keep up with what is going on. Yes.
Actually, this is great to hear this from you, since personally, me and Rimon, who asked this question, we did the same. We are moving from academia to industry, and it's very assuring how, yes, exactly. So thank you.
And you know that you have to have so many skills, and then you just realize, oh, I have it. You know, so that's also important, because you have to be at the beginning, and you know, a business person, have to be the science, and then you are responsible for all of your employees to make sure that, you know, they maintain there, and then you have to guide them and rely on their work, and so it is great thing. Congratulations.
Yeah. Thank you. We are very much enjoying it. Yeah. So, yeah, hopefully we'll have like a new product, a new innovation, and thus we will really feel the impact. Mm-hmm. So, yeah, thank you. Okay. So the question after.
There's Islam Farag said, what are the learning sources about mRNA therapy? Learning. So where you can learn. I mean, I put all of the references on my presentation. If you know, you want to read older papers, I have to tell you that older papers might have less data, but what they describe is a lot of thinking, more thoughts in it. I love reading, even from the 50s. I love reading old papers, and I have so much respect for all of those people who came before us. And so there are a lot of you know, sources that you can read and learn. And I just showed the, you know, the recent review. And if you cannot have access, you know, just email me and or to you first, and then I can send any papers that you would like to read.
So is there a prepared lab for messenger RNA targeted therapy or experiment in Egypt? Actually, I'm not aware of any lab working in messenger RNA in Egypt. We are planning to do this at MARC, but not yet.
So for that one, I can tell you, Samira, that in 2013, we created International mRNA Health Conference. This is a non-profit, so we are doing as volunteers, selecting a presentation and so on, and try to bring together the whole wide world. So there are RNA companies in Korea, in Japan, Iran, scientists also applied for posters, and then these meetings, this year, it always in November, and the first part, and then in this year it will be Boston, US, and the other year is Berlin, Boston, Berlin, so that you know, from the area students can come, and the students are paying nominal fee. It is also accessible online. You can watch all of the lectures there, and we don't make any profit, and many sponsor is enough that who even show up, you know, and we also give out stipend for travel so that you can meet those scientists who are on the field. And so it will be on November 7 or something this year, 8 in Boston, and it is the 10th already.
Okay, great. It's great to know this. Yeah. So the question after that, why lipid nanoparticles are chosen as preferred delivery system for mRNA?
Which one is that?
By Lamia, why lipid nanoparticles are chosen as a preferred delivery system for mRNA?
Mm-hmm. Yeah. So when I mentioned that there were liposome, 1965, when scientists in London discovered. So the liposome is contains negatively charged, those are phospholipids, and the RNA is negative, the lipid is negative when you try to make the whole thing, so it is very diluted. So you have a big unilamellar liposome and a little material in it. Now that they come up with the positively charged lipid, yeah, the lipofectin. What happened is in vivo is very toxic, because immediately the RNA complex with this positively charged, it sticks to the albumin and others, and problematic. So what is the lipid nanoparticle? What scientists in Vancouver discovered that if you have a positively charged lipid, but it is positive only if you have very low pH, so that what they do is the pH they make low, and then the RNA negative, the lipid is positive. When they bring it up to neutral, it will be neutral now. But now that they add additional lipids like pegylated lipid, cholesterol, and others, and so what they have at the end is very packed. A lot of RNA could be incorporated to such particle. And what I have to say that the lipid nanoparticle, you are asking, first of all, to protect the RNA when it is in the circulation, help to enter to the cell. When it enter into the endosome, then you want this particle, please release the RNA to the cytoplasm. And what we also ask, that please work as an adjuvant, because what turned out in the present vaccine, the ionizable lipid, this lipid which I told you that you change the pH and then it will be changed the charge. What happened is this is the adjuvant. That is stimulate the immune system to recognize the protein translated from the messenger RNA.
So the question after that, are there available protocol for designation?
What designation means?
Designation. I don't understand. If you can elaborate the question again, please.
Yeah, for some.
Okay.
Mm-hmm.
Yeah. So the question after, have you used mass spec to determine degradation of lipid nanoparticle and or short interfering RNA? And what instrumentation has BioNTech uses for this?
Of course, everything was used for the vaccine, mass spec, and many other things. I don't know that the degradation of the virus. So that I think it was, we spent a lot of time to invent assays, that what we needed. And some, I don't know, 55,000 or whatever assay had to be performed. And then with the composition of the lipids were also mass spec analyzed and determined. And not, not what we had the instrumentation. Many was done by contact organization and also by Pfizer. But we also did a lot of toxicology and other study. All of them is available. All of the information is publicly available.
Okay, I think Dr. Samira lost connection, so I'm gonna continue the questions.
Please, here
Dr.
is
Samar
one.
El-Achi.
I read one here, Joseph Pools. Please, what is the main difference between self-amplified, self-replicating RNA and mRNA in perspective to in vitro mechanism of action? Do you think that siRNA one day would dominate over mRNA? So that we did our phase 1 clinical trial included self-amplifying RNA for the COVID vaccine, and but it was inferior. So it seems like it works quite well in mice and smaller animals, but it might not as effective. Especially one problem is that it was inducing interferon, because there is a replicative intermediate, even if it is replicating, or the RNA, double-stranded RNA is usually in the spherules, is hidden, but still it induces interferon, and the interferon is interfering with antibody production. It had been shown by Locke that and others that the interferon is inhibiting CXCR5 expression on the follicular T helper cells. And who are those T follicular helper cells? Those are the cells which will go in the germinal center and help the B cells to produce better and better antibodies. And that is interfering here. And so that's maybe self-amplifying RNA could be used for cancer vaccine, where only the cellular immunity is important, but for infectious disease, best to my knowledge, is that is challenging.
Okay, thank you so much, Doctor. Let's move to the next question.
Can the mRNA treat immunological disease such as rheumatoid arthritis? Yes, that would be, there is a chance for it, because this is an autoimmune disease, and maybe similarly we could induce tolerization, but we have not done experiment for that. For that, you also, it's challenging everything for the cancer to identify the cancer antigens, because in the case of coronavirus, it was obvious that on the surface there is this spike protein, so that has to be encoded by the vaccine. And that's why the reason that Moderna and BioNTech, Pfizer, all of them, 100% identity of those amino acid composition of those vaccine, which, or the protein which coded by the RNA, the amino acid is the same. But of course, for other things, is that you have to identify what could be what should be encoded, like in an autoantigen, what could be it, or in the case of cancer, what should be the cancer, what is the cancer antigen, and even if it is generated, whether it is the driver mutation, so whether you can eliminate. So there is more challenges there, and but just like we learned so much those 20-30 years of working to develop a vaccine for HIV, then maybe eventually we will learn soon. Actually, next summer, those phase 1 trial will end, at what Moderna describing. So we will learn that whether that other big pandemic, when I arrived to the United States in 1985, that was a serious problem, the HIV. And considering that for years, even detection of the virus was not available, that, they couldn't culture, couldn't make tests. So, the advancement of molecular biology helped a lot. Are there relation called the Abdel Halem SAD? Are there relation between personalized medicine and mRNA therapy? Yes, there are mRNA-based personalized medicine when BioNTech is performing such a clinical trial with Genentech, where patient sample is collected, sequenced, and identify those proteins which had mutation, and then mRNA coding for those so-called neoepitopes are generated and injected to the patient. And so each patient had their unique vaccine. So it is in use. And also other companies has similar program. Peter Macron, in case of commercial approved mRNA vaccine for the COVID-19, the spike protein code has been modified in several ways to enhance activity in vivo. One of these modifications was the 2-proline amino acid substitution so that the protein can be translated in the proper structure, be active. The question, how can we know that amino acid to substitute if we are developing other therapies that code for proteins that are not naturally translated in vivo? How can we know? So, it was the same, actually, not just for the COVID-19, the HIV I just mentioned, because what they did realize that if they have individually the GP160, or whether they are 3 of them together, so they eventually link together, which is naturally not linked, but they had to make an adjustment based on knowledge of the 3-dimensional structure of those proteins. So first you are trying with an unknown protein to code for whatever sequence is available, and whether others already have crystal structure, or realizing that the molecule different form is active or inactive, then had to be adjusted. So how can we reduce side effects of mRNA COVID-19 vaccine? Amira Ezzat. So Amira, yes, there are side effects, and one of them is, which was identified, is to have inflammation in the heart for especially young boys. And of course, it could be related to the protein itself, because the virus is causing a similar, much more serious problem in the heart, and even arrhythmias, which actually the vaccine is not causing. And other effect, you have to realize that the vaccine is injected to everybody. So when you have a medication, usually you have the clinical testing, identifying, and or what other additional disease somebody is suffering, and then, but here you don't have that. And of course, you can see that, oh, people died. Yes, people died before there were any vaccine. 10 years ago, 100 years ago, they died, just all of a sudden. And so what they were doing is, the recent study has identified that what is the different disease by different age, which looked at 1 million person that gets in 1 year after getting the vaccine, or 1 year after not getting the vaccine. So those people who never get the vaccine. And when they compare, they found the same number of people get stroke, get other heart attack and other things, and only this heart inflammation was significantly different. And so, you know, the reports for the side effect, those are scientists and physicians are looking at and see that whether it is a direct correlation, whether it is just association, and we'll investigate. And there are more and more publication came out showing case reports showing that the patient had some kind of ongoing immune reaction, and then the vaccination resulted that it gets stronger, that reaction. I have to tell you that in the same number of people who said that they had never had joint pain and now they have, and the same people who were saying that they have always had joint pain and they get the vaccine, now they don't have. I also have even a letter from somebody who wrote that his mother was Alzheimer and for 2 years didn't say a word. And after the second dose, she started to talk again and responded normally. So what happened is, and another interesting thing I may mention, the side effects, is that I received the head of the hospital who treating people who did not cannot conceive, and she said that after vaccination, it seemed that those couples could get conceived again, and then she wanted to know that what I think about. So in 2004, actually, I was writing a review about that when there is a inflammation, then if there is an inflammation ongoing, things get much stronger, but after a certain time, couple of days, what happened is that there is an immune suppression, as it is very well known, those people who get operated and everything went fine, but of course the operation is a big insult, and what happened is that the patient died of an infection, a lung infection, because by that time, as a consequence of big insult, the inflammatory insult inflicted by the surgery, it created an immunosuppressed state and then did not respond. So what I think is that in certain cases, when beneficial effect was mentioned, like when inflammatory process was broken and then it was eliminated, very likely this immunosuppression was helpful. Immune activation could be also helpful. Many people said different viral infection disappeared from their body, but there are also when people had some kind of inflammatory reaction and it made it worse. So it's obvious that the vaccine is not inert. You have an adjuvant any time, any vaccine, and were also studies, which they complain to me that, their menstruation cycle was interrupted. Actually, 100 years ago, it was already reported that other vaccines also did that. But it never happened that 1 billion people would get the vaccine at the same time. And then they would say, oh, did you get that? I get that, because they gave to children or partially, and not everybody. So that's also a component. But obviously, there are other unwanted effects are there. Okay.
So I don't think we have any more open questions. That's okay. Right. Yeah. Good. Thank you very much, actually, for your time and effort. And it was really great discussion and presentation and everything. We appreciate it very much.
Yes, I just can tell you that I not mentioned in the beginning, but, I came from a small town. My parents were not formally educated. They have elementary education. And if you put your mind into it, learn and not complain, do it, you can reach anything. I mean, for me, is not, recognition is what I got today. The joy that working in the laboratory, solving problems, thinking this, thinking that, and advancing things, that's the real thing. And then I also might mention that when I was in high school, I was reading a book. Hans Scheyer wrote this, who was Hungarian, born in Hungary, so they translated in Hungarian. And what happened is that he described, actually he used the word the stress first for the human feeling, not for the physics. And what was there is that he said that, that you are paying too much attention if you look at, oh, my colleague, it more advanced, he get more promotion, more money. You already took away your attention, what you can change, because you cannot change that. And just focus on what you can do. And that's also another message was that he's saying that the stress can kill you. And not really the stress, but how you perceive. And he recommended that you learn to convert the negative stress to positive. Because what he said, without stress, life is not important. You won't get up in the morning if you don't have this anticipation, excitement, because all of this is stress. And so that's what I could suggest also to the people there, that I was terminated in my position several times. And instead of focusing on why me, I worked so hard, why they send me out, away, I focused on, okay, now what I can do. And so all of this energy put on something that you can change. And this is how I get here to the BioNTech, because even less than 10 years ago, I was shown the door. Okay, so think about that. Nothing is tragedy that you sent away. It doesn't mean that you are not good. It is just, you couldn't express properly yourself, or that's what I tried always think, that what I can do, and instead of blaming others, because that won't help you. So if that positive, and I can say, and I also, to the women, and I am so glad that you girls, women are here, would say that my message is that you do not have to change. You do not have to decide whether I should have my career, whether I should have a child, because really you find a supportive, good husband, then he will realize your dreams and support you. And I have also a daughter. I didn't help her with the morning dressing up other things, and she turned out fine. She's a 2-time Olympic champion, and she's actually working a company to make cap for the vaccine in San Diego. So everything is fine. And so with that, I wish you all of the best.
Thank you for these inspirational, motivational words at the end of our webinar. So enjoy the rest of the night, and thank you, everyone.
Yeah, make sure that I didn't say anything to the guys. I can send a message to them. Be a good husband.
Yes.
And have your wife.
Yes. Actually, today is the Egyptian Mother Day, so your talk in the Egyptian Mother Day is perfect. So thank you very much.
Thank you very much. Goodbye.
You, everyone. Bye-bye.
Thank you so much, Dr. Katalin. Okay, goodbye.
Bye.